Okay, welcome to Genetics, Chapter 4, Sections 13 and 14, and the special topics on epigenetics, Lecture Part 1. This will be Part 1 of 3 parts recorded and posted on YouTube. All right, so now we're going to talk about whether phenotype results from genotype. So there are different genetic and environmental factors that will either promote or inhibit expression of genotype to phenotype. So just because you have a genotype doesn't mean you're going to have the phenotype. Now we're going to turn a lot of that beautiful, simple Mendelian P color stuff on its head. Okay, so there are genetic factors we're going to go over first. Then environmental factors we'll go over probably in the third part that will affect when something's expressed, how strongly it's expressed, how many things. Okay, so multiple conditions can affect when and which genotype will lead to phenotype that can be changed or blocked. Okay, so let's move on. So there's some ideas we have to go over. So penetrance is something we've talked about a little bit before. Okay, so penetrance is, got changed to the pen. Percent of genotypes that show phenotype. Okay. Recall with phenylketonuria or PKU that with diet intervention, even all of those who have the 2 recessive alleles for PKU have 0% penetrance. In other words, they don't show the phenotype, even though they have the genotype. Okay, so that's an example of penetrance. Expressivity is a range of expression of a phenotype. And we'll go over some examples of both of these. Okay, so first example is polydactyly. So you see this image here. I know it's kind of creepy. So polydactyly is the presence of extra digits, whether they're on feet or on your hands, fingers or toes. Okay. So for individuals that are have one dominant allele, notice that polydactyly is a dominant trait. Some that are P whatever are not. They don't have polydactyly. Okay. Button on accident. They're not polydactyly. There's a range of expression in these individuals. So the size and number of extra digits varies. Some people have just a really small, one little small partial digit. Some have complete digits. You can see in this individual, I had to count. There's 3 extra digits on both sides. Okay, so polydactyly shows both expressivity and penetrance. Some who have the dominant P allele are not, they don't have extra digits. And then there's a range. So the polydactyly gene and individuals that have one dominant allele, there's differences in penetrance and expressivity. Okay, with Fragile X syndrome, we're going to talk more about this later. This is a dominant disorder, dominantly inherited. Okay, it's the most common form of inherited mental disability that we know of. We're going to go over the cause a little bit more later, but Fragile X syndrome is there are fragile sites on the X chromosome. So the penetrance is 30% in females versus 80% in males. Okay, so I want you to take a second to think about this. Why are more males affected by Fragile X than females? Pause if you still don't have an answer. Because how many X's do females have? They have 2 X's. Males only have 1 X. So if they have one Fragile X, they're going to be more susceptible to the issues with these. And so Fragile X is there are fragile sites that don't stain well, that cause weak points that cause the X chromosome to break. Okay. There's also a range of mental disability. So in other words, there are expressivity differences too. Differences as well. So Fragile X syndrome also has varying penetrance and expressivity. Okay, so now we're going to look at the eyeless gene in Drosophila. My head. All right, so all of these images have the same genotype. All same genotype. What? Okay, nice normal eyeball. No eyeball. Okay. In the lab, you can control the genetic background and the environment. Okay. And so we know based on those studies that the genetic background and the environment can affect when genotype of eyeless goes to phenotype. So because of lab work, in a lab we can completely isolate both genetic background and the environment. We know that genetic background, that's my background, and the environment can affect how the eyeless, oops, I missed an E, genotype leads to phenotype. Okay, so we're going to start with genetic background. So what are the genetic factors that can affect how genotype leads to phenotype? So we're going to move on to the meat of the genetic factors portion. Okay, so there are 3 basic genetic factors or things to consider that can affect whether a genotype is expressed as a phenotype. One is genetic background, which stands out of reason, genetic anticipation, and epigenetics. Okay, so 3 different ones. So we're going to start with genetic background. We'll define this again, but this is basically other genes. Okay, so other genes can affect the expression of genes. All right. So this is why we use identical genetic clones. This is why identical genetic clones are used in research, with one small difference, hopefully, to see the role on the same genetic background. Okay, so that helps to account and control for this. That's why all the flies that you're using, they are true breeding for everything. So they are basically, like all the wild types, are clones of each other, and the same genetic background is on your mutant flies except for those mutations. So that's how we can do these genetic studies that we do with the flies. We do the same kind of concept for mice. Okay. All right, we're going to go over more details on genetic background in a few moments. Then we have genetic anticipation, and basically that there's an increase in severity of the phenotype with each generation. It's kind of sad. And then epigenetics is above the gene inheritance. So things that you can inherit that are not the AGCT, okay? Has to do with 2 main things. Number 1 is the state of chromatin packing. Okay, what's chromatin again? All right, that's the DNA with the protein, the histones, okay, and that equals chromatin. So how tightly or loosely is chromatin packed? Obviously, the more tight it is, the harder it will be to express the gene. The more loose it is, the easier it would be to express the DNA. And then there are both DNA and histone modifications that can alter expression of genes, that alter gene expression. And some of these modifications have been shown to be heritable. All right. So we're going to go to number 1 here. Oops, wrong button. On genetic background and start there. Next. Okay. So genetic background, we already said, is all the other genes' DNA, right? So all other the genes and DNA, basically, because it's not always genes. Okay, many mechanisms. We're going to go over 4 different mechanisms. Okay, 3 are on this slide. Okay, so 1 is what's called copy number variations. So copy number variations are extra copies of genes. You can imagine just having extra copies of genes can alter how much of a product is expressed, right? You have 10 copies of a gene, you express the gene, you get a certain amount of protein, and then let's say now there's 20 copies of the gene. Stands to reason you should have more of that protein made, which might be a good thing, might be a bad thing, may not change anything. Okay? So, copy number variation has been shown to be related to 10 to 20% of autistic cases. Okay? 10 to 20% of autistic cases. All right, if you look at the SFARI, I forget what it stands for. It's a database on all things genetic and autism. All right, I looked at that database, and the number of copy number variations, there are 2,174 CNVs that are associated with autism. Those are CNV loci. Okay. So there are a lot of different places where there's extra copies of genes that have been associated. It doesn't mean they cause autism, but they're associated with autism. Okay. Another example is HIV suppression. Okay. There's a gene, the CCL3L1 gene, which codes for the MIP1 alpha protein. Okay, that protein is a pro-inflammatory molecule. Protein, and the more copies, the better HIV suppression, or the better the HIV host insusceptibility. So we'll say HIV resistance. Okay. Okay, there's other examples, but those are 2. So copy number variations, the more copies you have of a gene, it can affect not just expression of other genes, but whole multifactorial conditions like autism. Okay, so now we're on to gene networks. So gene networks, for example, there are cancer gene networks. So we're going to go over, there are proto-oncogenes. So these are genes that promote cell cycle. Now that doesn't mean they're bad genes. When they're working properly, we need to promote the cell cycle when there's damaged tissue, we need to replace it, or when children are growing. It's kind of nice for cells to grow so they can grow. Okay. And there are tumor suppressors that inhibit cell cycle. Okay. And those are good as well. All right. So a mutation may not lead to cancer, but sometimes it can, or multiple together. So we'll think about these 2 later on. Okay. And the last on this page of the 4 different genetic background examples is polygenes. So with polygenes, multiple genes, wow, I'm not writing very well right now, are additive to lead to the phenotype. Okay. Examples are height and weight. For height, each gene only allows for a few millimeters in height. So if you lose one, it's kind of unnoticeable. Okay. So moving on to the 4th example of genetic background and how genetic background can affect expression of other genes, and that is position effect. Okay, which basically means where a gene is located matters. All right, so position effect is change in expression, okay, of a gene due to a change in the genome position. And so my question to you is how? Okay, how can you move? So if you move an intact gene somewhere else, okay, the coding region is moved. What's something that could easily happen? What's other pieces of DNA around that you could separate from that would cause an issue? maybe we just take the coding region and now we are farther from our promoter. It's one thing that can happen. So separation from promoter. From the promoter sequence. Would that enhance or inhibit expression? So the plus means enhance, the minus means inhibit. So if you take the coding region and you remove it from the promoter region, that gets the whole transcription party together, what's going to happen? We are going to inhibit. Okay. Less likely, but possible, is a gene is repositioned with a different promoter. Okay? So will that be inhibitory or stimulatory? Think about it for a second. Do we know for sure? No. Maybe it's repositioned next to a stronger promoter, which would stimulate expression, or maybe it's repositioned next to a weaker promoter, which would decrease the expression. So it depends on the original promoter and the new promoter. Okay? Finally, you can have a gene moved. Oops, I forgot to change it back to blue. Let's keep this moved near what it's called heterochromatin. So what is heterochromatin? Heterochromatin are areas of condensed chromatin that is genetically inert. In other words, dead. Doesn't do anything. Just lies there being DNA. And heterochromatin will inhibit nearby gene expression. So it stands to reason if you move a gene near heterochromatin, what's going to happen? Are we going to stimulate or inhibit expression? We're going to inhibit it. Okay. So an example of position effect that can actually has been associated with certain cancers, okay, where an allele has been separated from a promoter. So my question to you now is, let me change the color so you can see it's different from the other. Was that allele for a tumor suppressor or a proto-oncogene? So a tumor suppressor inhibits the cell cycle. A proto-oncogene stimulates the cell cycle. Which one, when it's separated from its promoter and is expressed less, would lead to cancer? Pause it and think about it. Did you pause it? It would be a tumor suppressor, of course, because a tumor suppressor, if it's not near a promoter, okay, it will be expressed less. And if there's less tumor suppressor, it'll make it easier for the cell cycle to happen. Okay. We're going to look at another example about eye color in female Drosophila, and that's on the next slide. So this example of position effect. Okay. So this is an individual. That's a fly. Okay. This is an individual heterozygous for a wild type and the white. Okay. The white eye, in this case, this is a recessive allele. Okay. And their normal eye color would then be red, right? Because you have one wild type allele, one recessive white allele, so this should be white. So if the positive allele, which is the X to the positive, is translocated near heterochromatin, the eye color ends up being variegated, which is a mix of red and white, even though this individual is X plus XW. This allele has been moved to heterochromatin, and it is no longer being expressed well, so it's expressed less, and so the white allele ends up being expressed more. And this would be the quote-unquote normal, this individual is X plus XW without that move. Okay? All right, so just let's recall where we are. We know that both genetic factors and environmental factors can affect when genotype goes to phenotype and how and when. All right, we're in the middle of genetic factors, and we just finished genetic background. Okay? So now we're going to move to genetic anticipation, number 2. So genetic anticipation applies to certain diseases, and basically it's when diseases get worse with each generation. So subsequent generations have an earlier onset of disease with increased severity. So that means if grandpa had it, it started later in life and it wasn't as bad as the grandson who got it earlier and it was a more severe phenotype. Okay? They are associated with what are called trinucleotide repeats. A trinucleotide is 3 nucleotides that's repeated over and over and over and over and over again. And there's an example below. Okay? There are more than normal. Okay? This can happen in germline cells, so in the sex stem cells, so passed on, or in somatic cells. Okay? So I'm going to write something down here. If repeats are in an exon of a gene, that'll cause a change in protein, and therefore probably a change in the function of the protein. Okay? If the repeats are near a gene, can change the expression of the gene. Okay? Maybe you make more or you make less of that. Okay? This can be changing to an activator region or an inhibitory region, promoters, etc. Okay, so that's what trinucleotide repeats are. All right, now we have 2 examples here of diseases that have genetic anticipation, where it does get worse with every generation. So myotonic dystrophy, when you say myo, that means muscle for my AMP people. There are 5 trinucleotide repeats, it's normal. If there are 50, there's minor disease, and that means they probably have some cataracts, maybe some mild muscle issues. There are more than 1,000. There's severe disease. This is often fatal after birth. Okay, so the more repeats you have, the worse the phenotype, the more severe the phenotype, and the earlier it sets on. Okay, Huntington disease and Fragile X also have genetic anticipation. So this example here, this is Huntington disease. Okay, so now you see CAG, 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 CAG. Okay, so in normal individuals that are wild type, quote unquote, there are 12 to 37 repeats. Okay, when that is expanded to at least 60, that's when you have Huntington's disease. Okay, and with Fragile X, you end up with more fragile sites each time. As inherited, it gets worse. All right, so a question for you. Why does increased repeats make chromatin unstable? Okay, so question. Why does an increase in repeats make chromatin unstable, especially with Fragile X? Or both of these, this is how this actually happens. It's actually an issue with replication of the DNA and DNA repair. So the more repeats there are, it's almost like if you had to write the word 1 100 times, and then you had to copy it again on the next page, it'd be easy not to write the exact same number of ones. And so there's some errors that occur. So each time the DNA is replicated, there's a good chance that it'll be expanded. And as these get expanded, and somatic cells, which are your normal cells, it can cause increased phenotype. And then if it's happening in germline cells, that's where it gets passed on, and the next generation has a worse phenotype because they have more repeats than the previous generation did. Okay, so now moving on to number 3, and we're just going to start epigenetics today and move on further in number 2 of these video series. Okay, so with epigenetics, this is above the gene. So in other words, it's not just the DNA promoters, enhancers, transcription factors, and all the other enzymes involved in gene expression, right? There are other things that can be modified that change gene expression besides all of that. Okay, so epigenetics is like a catch-all term for various processes that regulate gene expression. Okay, so that's what epigenetics is. So purists require heritability. Okay, so purists would define epigenetics as a heritable change. Oop, I'm not sure what I'm hitting. Change in phenotype without a change in the nucleotide sequence. And you're probably thinking, what? Like, that's crazy. Okay, so there are a lot of different epigenetic mechanisms, so to speak. One we're going to talk about today is DNA methylation. Okay. Two we'll talk about later is chromatin remodeling. And we've kind of hinted at that. Chromatin is, again, the DNA and histones together. How tightly or loosely the chromatin is packed will affect gene expression. Okay. 3 is microRNA. RNAs are so many of them. Then we're going to talk a little bit about long non-coding RNA. Not just tRNA and mRNA anymore. 5 are prions. Okay, so prions we're going to define a little bit here, and we're not going to get back to them again. So prions are proteins that are capable of self-perpetuating. Okay, so they're proteins capable of self-perpetuating. Changes in protein shape. So basically one protein can change, and it could lead to another protein of the same shape to change likewise. And that, of course, if we change the shape of a protein, we change its function. An example of this is the SW1, yeast chromatin remodeling transcription contest. So this is a lot. So yeast chromatin remodeling transcription factor complex. Okay. This is inherited cytoplasmically. This is inherited cytoplasmically. That's crazy. Okay. So it's protein-only inheritance. It's part of a complex that regulates 6% of yeast gene expression. So it's slightly important. 6% is a lot. Okay, so this is an overview of epigenetics, and we're going to spend a little bit of time on DNA methylation before I sign off for this part 1. So DNA methylation is adding a methyl group to DNA. So CH3 is a methyl group. That's a carbon with 3 hydrogens bound via covalent bonds, and they are always added to cytosines. Okay, by an enzyme called methyltransferase. So methyltransferases are enzymes that add methyl groups to cytosines on DNA. Okay, and usually it's a cytosine next to a G, and these are called CpG dinucleotides. Okay. Methyl added to DNA inhibits gene activity. Okay, so what happens is the methyl group occupies a major groove of the DNA molecule and blocks transcription factor binding. And if we block the transcription factors, therefore we inhibit transcription and expression of that gene. Okay, I'm going to post something soon with some YouTube videos to show some animations of these that I would show you in class, but they're not easy to integrate into this recording platform or app. Okay. So on this figure, all right, you see a couple things. So on the top here, okay, so here's a CpG island. So there's a CG, 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 a bunch of those together, okay? And each one of these represents a C where there is no methylation. So these clear circles up here indicate there's no methyls. Okay, so this is, I'm going to change the highlighter. Oops, do I have a highlighter? Unmethylated. Okay, and it can be transcribed. Okay, so there's no methylation here. That doesn't mean the gene is on. You still need to have transcription factors. The chromatin still needs to be open. There needs to be no inhibitors bound to silencer sequences, etc. So there's still a lot of other things that have to be in place. But the DNA itself, since it's not methylated, it will not inhibit or not prohibit transcription if transcription is probably going to happen based on the other things that are around. Okay? Now we come down to B on this image. All right, and the promoter is methylated. Okay, so it's a CpG island in the promoter. And notice that all of these are black. So that means on each one of these, you have a CH3, CH3. I'm not going to write all of them, okay? CH3. So those are all methylated. And that means, change it back to blue, that a transcription factor is going to try to bind, but it can't. Okay? So if a transcription factor can't bind, you can't transcribe it no matter what else is going on. So there can be transcription factors, chromatin can be open, there's no inhibitors around, and because it's methylated, we're not going to express it. Okay? So even, oh, it's blue. Even if it's methylated, even if all the stuff around the gene is silenced. Okay? All right. So that's how that works, and I'm going to look for an animation for you to look at. All right. A few more things about methylation before I set you free for video number 1. All right, is that allows for tissue-specific expression. Okay, and so let's talk a little bit about that. So that means, oops, go away. All right, so we're gonna kind of describe that a little bit. So we're talking about differentiation. So DNA methylation is part of the differentiation process. What's differentiation? That's going from an unspecialized, more stem cell-like cell to a more specialized with specific jobs cell. Okay. For example, it helps a liver cell turn off genes not needed. Turn off genes not needed for normal liver cell function. Okay? This works with, so it's not the only thing, chromatin packing and histone modifications as well. Okay, so DNA methylation is part of how differentiation works. And it makes sense. Differentiation is a beautiful process because we don't need to express the entire library of genes. We only want to express the genes that we need to do our job as liver cell or a kidney cell or a wing cell on a Drosophila, whatever it is. Okay? DNA methylation is heritable over generations. I might send you some paper links to look at some actual data on this, but there's strong evidence that methylation can be heritable from one generation to another generation. We'll go over some examples in class 2. We also know that methylation can be essential for learning and memory in mice, and there are more studies. What they did in these mice is they inhibited methyltransferases. Okay, so they didn't allow methyltransferases to work, and that inhibited the mice from forming new memories. And so it's not just differentiation that methylation helps with. Okay, it's also been shown to be important for learning and memory in mice, and there are other studies supporting that as well. And now we're going to move and preview parental imprinting. Okay, and this is only something in mammals and angiosperms. Basically, it's a sex-specific pattern of methylation, okay? It's kind of crazy. It's going to be hard to explain. So this is where I'm going to stop video 1. We'll move on in video 2. We'll review methylation and dive into parental imprinting. I wish I could see your faces when I go over this. Maybe send me your faces as you watch the video. All right, you guys. God bless, and see you in part 2.